All right. Well, thank you, everyone, and thank you for the organizers for having us. This is actually Stellaromics' first podium presentation at a conference. So everyone in the room, you are now at the cutting edge of 3D spatial biology. So Stellaromics, we have our three-dimensional theme, and we're helping the world see biology in all dimensions. And the talk for today is the next dimension of discovery, advancing spatial biology in 3D. And this is really meant to inspire what you can do in 3D. I don't have enough time to get into every single detail. We do also have a booth where we're kind of in the center of the vendor area and would love to talk more. I'm Jay Clark, leading sales at Stellaromics. And I actually joined the company last year and had the privilege of helping build the technology with the team. And now we're rolling it out to the research community, which is quite exciting. So we've probably seen a different version of this slide many times today, but what I'm going to focus on is where spatial biology is going. So when we think about this year, 2025, and if we think about 2030, are we going to be looking at thin tissue sections, or are we going to be wanting to go into 3D tissue sections where we're actually seeing that true spatial heterogeneity? Obviously, we think that we're going into the 3D direction. And if all things were equal and you could choose between 2D and 3D, you would want to have that true intact tissue that you can work with. So that's why the company has been formed, and that's why we're building out this Pyxa platform, which I'll share more about. But just to set the stage, there's obviously the amazing use cases for the current platforms today that we've seen, but there are known limitations. I don't think these are too controversial, where the first thing that we do today is we take a thin section out of our 3D tissue block. And then you have issues with cell segmentation that's been brought up quite often, where if you happen to have a cell that's above or below your actual cell that you're segmenting, you might have overlap from those transcripts from those cells that are either at the top or the bottom. So it doesn't matter how good your 2D segmentation is, you always have that issue. The loss of rare cells and transcript, that's been covered quite a bit already. And then just thinking about the first step of your protocol, which is taking that thin tissue section. If you're looking for infiltrating lymphocytes or some rare cell populations, and you just happen to slice right above or below where you're looking for that feature, you're going to lose those data. And then lastly, everything today is looking at either mRNA or protein, but we're also building out an assay to look at that in-between step of RNA translation into protein. And I'll share a bit more about that. And just to put that point in a bit more of a visual, if we think about what's being done today, this would be an example of a 5-micron section where you have that single layer, these cells are cut in half, and it's a much more sparse data set because we're only in that 2D space. Versus where we're going, which is in the 3D. So this is a very small snapshot of some 100-micron thick mouse brain data that has been generated. And you can start to see now we're getting multiple cell layers. We get to see that cell-cell communication in 3D space. We're segmenting cells in 3D, and we have much more dense cell populations intuitively because we're going into the third dimension. And I'll actually show some of updated software that I think looks a lot better than even this bit later on. So that's all setting up why the company exists and what we're doing. The platform that we're creating to allow for this is called the Pyxa system. So this is the actual instrument here. You can't see my cursor, that's okay. So this is the actual instrument, the Pyxa platform. We're doing the whole integrated workflow with the reagents, the instrument that you see, and then also associated software that runs on the instrument. So what does this do? At the top level, this is the only way to get high-plex 3D spatial transcriptomic data today. So there's lots of ways where you can get thousands of genes in 2D, or you could do something like light sheet imaging to do 3D proteomics at a small scale, 4 or 5 markers. But we're currently in the range of doing anywhere from 200 to 300 genes in 3D, and we'll be expanding out from there. So that's the flagship assay. We're also building out RiboMap, which I'll say is really, let's say, a pharma-focused assay for us, where you're able to actively enrich for, or I should say, enrich for genes that are being actively translated by a ribosome. So we actually have an assay that can enrich for the ribosome, therefore taking us one step closer in that dogma of biology. And we see this as a really interesting application for groups that are introducing, say, different payloads into a cell, and you want to know, is my payload getting into the cell, but also is it being actively translated once it gets in there, offering a measure of translational efficiency in situ. This is also the first confocal imager on the market. So there's a confocal in here with a lot of new optics innovations that I can't share quite yet. But what this offers is that subcellular resolution at that 3D level. So we're looking at things like guide RNAs, small RNAs, even within the cell itself, because we're segmenting out that 3D cell. With that, the chemistry is quite flexible. So we're working in a variety of tissues, mouse, human, primate. We even have a group that's working on plants, just as an example there. So there's a lot of customization that can be done. And the last thing I'll mention, this is a very high throughput instrument. You can see it as a standing model, but with that comes a lot of throughput where if you took the same 2D area and did that in 3D, you're generating 10 times more data than you would with 2D. And what we're building is that you could do that in the same time to do that 2D data versus 3D with us. So just a really quick workflow. We have this as a detailed infographic that you can take home with you downstairs. But just going through this briefly, step one is section your tissue. We're doing this into a well plate format. And the difference is we're just sectioning thicker tissue. So we do 20 micron, 50 micron, 100 micron thick tissue, depending on the project. And we fix that tissue into a well plate. From there, we have these probes. They're called snail probes, kind of look like a snail. And these are meant to penetrate thick tissue and hybridize to your target regions. You might be able to see there's a little gray barcode on the probes themselves. That's where we're going to decode at the end of the protocol. From there, we do an amplification step, embed those amplicons into a 3D hydrogel matrix, and clear the tissue. So the end result of the protocol is amplicons that have barcodes linked back to the original RNA targets within optically clear tissue. So this gives us a very mechanically stable unit to work with. So even we've had folks that ship samples after the sample preparation process internationally, and then we've just imaged in our own lab because it is quite stable. And this also gives us the ability to do this 3D imaging over a period of a few days, which is what happens here. So essentially, the Pyxa platform is a 3D in situ sequencer. We're doing sequencing in situ. We can go into more details later on, but essentially what we're doing is with each 3D imaging cycle, we add another fluorescent nucleotide to the barcode sequence that is on that amplicon. And over the course of 3 or 4 or 5 cycles, we can decode those barcodes in the 3D space, process all of that data on the instrument itself, and then the output is going to be what is actually quite familiar to a lot of folks, which is going to be your cell by gene matrix. The difference is we're doing that with X, Y, and Z coordinates. So we're adding that third dimension along with all of the 3D images that come off of the platform. So we're actually doing the processing and the imaging live at the same time. We've got a pretty beefy computer on this instrument, stuffed with a lot of GPUs. If anyone wants to talk about all this tech details, I'd always have fun with that. And then from there, there's a variety of different applications and analyses that you could do. But everything is in 3D, right? We're actually looking at cell-cell communication in 3D space. So always get the question what this works on. Right now, we've been doing a variety of tissue types. We say this works with any clearable tissue. We had conversations with even folks doing bone and plants, which are very difficult to clear, but can be done. Primarily, we've been working with brain and retina samples to start, and most recently working with things like kidney, tumor, and even very difficult to work with samples like liver. So if you have a tissue type that's not on here, please come and talk to us. We're at that stage of a company where we're now working on a variety of different tissues. And I should mention for this group, we're primarily working with fresh frozen OCT embedded tissue, some PFA tissue, and then FFP is on the roadmap for next year. So again, happy to get that ball rolling for any of the FFP folks in the crowd. So this is a whole talk in of itself of what you do with 3D, but I'm just calling out a few examples of in neuroscience, you could reveal these short and long range connections. In oncology, you could think about the 3-dimensional tumor microenvironment versus the 2D tumor microenvironment, look for those rare TILs. Immunology, looking for those cell types and their spatial organization in 3D. And then I'd say one of my favorite assays is thinking about cell and gene therapy. Again, where you can introduce some sort of payload into a cell, you can visualize that within the 3D cell and look at the 3D microenvironment all at the same time. So here's some very quick examples, and I'll give 2 very short vignettes, and happy to go into more details later. So this is just an example of what 2D can miss in neuroscience. And again, the output of our platform is going to be these cell type maps in 3D, which you're seeing here. You have all of your neuronal and non-neuronal populations with its 3D maps. But if you think about a core spatial assay, which is nearest neighbor distances, if we look in 2D, what we're seeing here is that these cells actually appear to be much farther apart than they are in reality. So the blue is looking at cells that are 40 microns or more apart. Versus once you go into 3D, which is on the left, actually there, you start to see that these interactions and these nearest neighbors are much more close than you would think in 2D. And intuitively, this makes sense. You have more cell layers to work with, so things are going to be in its native 3D state. But this is to say that you're getting this different result in 3D than you would in 2D, that 2D might be misrepresenting those nearest neighbor differences. And then similarly, in a tumor example, we start to see these 3D tumor amplicons here, just as an example. This is a skin cancer where this mechanisms of immunotherapy response is not very well understood. And a similar panel was run. This was 250 genes. And we're looking at another spatial question, which is how much frequently are these cells next to each other? How often is a tumor cell close to an immune cell? And what you'll see here is in 2D, you see this interactome of cell-cell adjacency. And once you go into 3D, we start to see that adjacency and those events of interaction start to increase. So again, because we have all of the layers of data, we're able to see much more events than you would in just the 2D alone. And with that, pivoting a little bit into the next assay we're working on, which is RiboMap. This is mapping translation in space. And what you'll see here is on the right, we have our snail probe that targets the mRNA. And we also add a third probe, which targets the ribosomal RNA. And when all three of these probes are simultaneously bound, we can then go down into the amplicon and probe detection. And what this does is enriches for that ribosome-bound mRNA, therefore, again, getting closer to that dogma. Why would you do this? One, antibodies are hard. So if you wanted to do 1,000-plex antibody panel, that's difficult, but you could much more easily make an RNA-based ribosome panel to do that type of screening for different RNA therapeutics. But I'd say that the most interesting piece is what I referenced earlier, where if you have a payload that you're introducing, you can see how that's being translated within the cell. And you could even run the RNA, the mRNA plus the RiboMap together to get that measure of translational efficiency within your cell populations. There's, of course, a lot of basic science RNA regulation questions that can be answered as well. So always happy to have those discussions. So with all that, that was all on the chemistry, and there's been a lot of talk of analysis. We looked at the market and didn't really see a really good 3D spatial platform for analysis. There's a lot of really good 2D platforms out there today, but we're a 3D company, so we ended up building this. So what I'm showing you here is a snapshot of the mouse brain with software that we call Pyxa Studio to allow that visualization of the actual 3D cells. So now we're starting to zoom in to the tissue, and you're going to start to see all of the transcripts pop up within the cell. So each different color is a different cell type, and even the transcripts are colored. And now we're going into the tissue within all of the 3 dimensions down to the subcellular level, and now we're coming back out of it. So this is just a quick snapshot. It's built to do a lot of the features that were mentioned in the 2D platforms. You could select for specific cell types. You could look at your UMAP side by side. You could look for different transcripts. But we really wanted to build this as that exploratory tool for folks once they get all of this amazing 3D data back to be able to investigate that quite quickly. So this is a software that's included for anyone that works with us via services or the instrument. And that leads me to where we are today. So we are launching this platform this year. We announced the technology at the AGBT conference. We've received the first 3 orders, and we'll actually be rolling out those instruments in an early access period with a 4th one that'll likely be coming soon. So we're doing this in a kind of a soft launch fashion where we're going to do the first 4 instruments, make sure that all goes well, and then we're doing the full commercial shipments at the end of the year. So if anyone is interested in the instruments, the end of the year would be the timeline that you could actually receive one of those first ones. And as of today, we have a lot that's available now. So we have up to 100 micron thick protocols. Depending on your project, you might want to do 50 micron or 20 micron or 100 micron, but that's our maximum that we're at today. The system itself can go up to 200 micron and will be going thicker and thicker as time goes on. We have a mouse brain panel that is available off the shelf. So for any neuroscientists, that's a really easy way to get started. But a lot of the work that we're doing now is custom. So we do custom gene panels all across all of those tissues that I mentioned, kidney, tumor, et cetera. The Pyxa Studio, the 3D visualization that is available now. So if anyone wants a demo, come on by. And we are also taking the first dip into proteomics, where we can look at things like protein aggregates at the same time as this transcriptome. So things like amyloid, where we have these nice aggregates, and we could say, in fact, how big is your amyloid plaque? Is it a large plaque or a small plaque? We can look at all of that in 3D space. And then coming soon, there's quite a bit, RiboMap, FFPE, more genes, thicker tissue, and then even thinking about StarMap Plus, which is enhanced protein co-detection. But I'll probably get the questions, I'll just say it right now. Right now, we're doing a combination of kind of like a cell pose and a Bazor, where we do the DAPI plus the transcript localization to do the cell segmentation in 3D. But actually just saw some really interesting data out of R&D where we're doing different membrane stains right now. So the idea is we get the membrane stain in 3D. And then closing up, thinking about what you could do. We have a services lab in Boston. We call it Technology Access Services. So it's kind of like our try before you buy pilot lab. If you have a project, we would just meet and discuss it. You ship your tissue blocks over to Boston. We have a couple of our scientists in the back that would actually process those data. We go through the data reports, show you that Pyxa Studio 3D software, and that takes anywhere from a month and a half to 2 months, depending on how much customization you need. So with that, I'll say thank you. If you have any questions, feel free to email me. I'll be around here this afternoon and tomorrow. Thanks, everyone.